Yeah. Good morning. Yeah, and I would love to be there with you. Yeah, in Kingston. Yeah, but unfortunately I'm in South Africa. But I thought about sending you this presentation. Yeah. Today what I'm gonna highlight to you is not just on virus evolution, but what we think that was really important for our output and research in South Africa, which it was about the organizational aspects, the transdisciplinary research, which generates some relatively high output and impact on COVID-19 genomics in South Africa. My name is Tulio de Oliveira, and I am the director of the Center for Epidemic Response Innovation, CERI, and I'm a professor of computational biology. Yeah. So, so this presentation is gonna have it should be around like 10 minutes each of the points, and we're gonna really highlight really our philosophy in responding of the pandemic, which involved a lot of preparing, yeah, and then following by response, communication, and activism. In after that, we did massive. Really, we trained thousands of medical staff and scientists in South Africa and Africa, and then only after that we focus on the scientific output, which was the last on our list. Yeah. So everything starts on Twitter. Yeah, of course, the Twitter, especially in the last few weeks, not the most popular tool in the world. But in end of 2020 or in 2019, yeah, that's when I personally note from tweets from both true colleagues and friends, yeah, Sir Jeremy Faraday, director of the Wellcome Trust, and Andrew Rambold. And that just to highlight that is this describing the first one 31st of December, the other one 1st of January, highlighting these unusual cases of viral pneumonia, no human-to-human transmission, linked to a fish market, patients, many business owners in the market. If 1 and 2 is true, then must be a repeat zoonose, but from what? Case linked to locations sound more like Legionella. So instead of really having fun in the end of the year, we are following that, and that we highlight, we take quite serious some of the scientific alarm from both Andrew and Jeremy. Yeah. Then we follow on the 3rd of January as 8 o'clock on January 3rd, a total of 44 patients with an expected diagnosis of viral pneumonia were found. Yeah, and that was a direct translation from a piece in Chinese. And Twitter stayed the main contact for scientific information. By the 10th of January, the first genome it is released. Is another colleague and friend of us, Edward Holmes, that did that in the name on behalf of Professor Yongzheng Zhang. Yeah, and that's when we really start. So now we have the first genome. It was released in GZA. It was also released in Virological, and gene bank. Now we can start working. So that's what our team does. Yeah, so we start working first on getting both the assays to sequence this virus, but also to get a phylogenetic typing tool and assembly tool, so that we could go from raw data, yeah, this means FASTQ that come from the sequence, to assemble to identification of the virus. And we release in the 22nd of January, we release our software called Genome Detective, yeah, that allow us to really go from producing data and analyze genomic data for this new virus. At that time, the virus was still called N2019. And that's what we do a lot in South Africa. So just to highlight, yeah, on 10th of January, that's when we see our first genome. On the 22nd, we launch our software application. On February, we start ordering reagents, yeah. And then on March, we have the first case in South Africa. We even sequence this first case in South Africa. We start really equipping the lab for high throughput sequencing with a lot of PPEs and primers. And then on the 23rd of March, the country is locked. Yeah. On April, we get involved in kind of a big hospital outbreak that was really, really important to South Africa, that shocked the country. I'm going to bring some information about that. In May, beginning of May, we start a massive training program with thousands of clinicians based on the report of this hospital outbreak on how to control nosocomial infections. And in June, we have already launched a network for genomic surveillance. This is 9 different labs in South Africa sequencing in real time. So as we move, let's just see where everything started for us. It is started on things that we have done a lot on the past. That is outbreak investigation, more specific nosocomial outbreaks in a hospital. On the 4th of April, we are asked by Professor Salim Abdul Karim, that the chair of our Ministerial Advisory Committee, short for MAC, to undertake an outbreak investigation. And at that time, only 16 cases and 4 deaths have been confirmed. And that's a direct approach that we are asked by Salim Abdul Karim, but more specific by our health minister to get involved on that. Why they ask us? Because we have participated in many nosocomial outbreaks before. One of them was a very famous one in Libya with both Oliver Pybos and Andrew Rumbold, but we have done many other ones, so we kind of knew what to do. So we moved to the hospital, and not only I take my infectious disease specialist teams, that medical doctors, that I specialize in infectious disease, and we start understanding how this nosocomial outbreak happening. Just to ground you here, in the y-axis I have the 16 first patients, or the 14 first patients, and the x-axis I have the dates, and then we start putting this puzzle together. So we had two potential start of this outbreak. One is someone that fly back to South Africa on the 7th of March. By the 8th of March, start developing symptoms. That's what that little dot it means. By the 9th of March, he presented at the emergency department of this hospital. We had a potential second source of this outbreak, someone that developed symptoms 9th of March overseas. Fly to South Africa and then presenting to the hospital at 11 and 12. But as you can see, what really happened, so we start putting all the patients, and here's the color code, the different wards that they went. So for example, this patient 3 become very crucial. You're going to notice soon. It starts in the emergency department, then move to the cardiac ICU. It has a heart attack, then stabilize it, go to the medical ward number 1. It's go home and then come back and then to the medical ICU, and then moving to the cardiac ICU again. So that's what we do for all the patients. It's kind of a detective work, and then we go in a little bit more details of each of the wards. So what we can see a really clear putative introduction of SARS-CoV-2 in the hospital. That's the patient that arrived at 9th of March at 17:45. He's screening at the entrance because he has symptoms. He's gonna do a test, and he's staying in the triage area. At that same time, this patient number 1, he get be in contact with 5 medical staffs. They do an assessment, and the nurse collect the sample from that. At that same time, around 15 minutes later, another patient arrived. Patient number 3 is an old lady that arrived with a cardiac arrest, of a stroke, and she's putting this bed to be able to be stabilized. And we really think that's how it started. That was a potential transmission from the first patient to this patient number 3, close proximity, either by direct contact or by fomites or by medical doctors and nurses. What's happened at patient number 3 around 13th of March is moved to the a medical ward, and then straight after that another patient in front in another bed tests positive. But what we notice in this medical ward, that was people that were hospitalized for other reasons, we have another 5 patients that test positive, 4 sharing a room, and another one here, which really suspecting that you have transmission not only by aerosol, but potentially by dirty medical equipment or medical doctors and nurses doing a bridge between them. Yeah. So we put all the timeline together. In the end, it's a very large outbreak. We can identify a lot of overlapping areas between the different patients, and we can start thinking that we can understand the steps of transmissions between the hospital. So as we get a look on the epidemiological curve, in the end we have 119 infections, which after start infecting patients, both in the emergency department, the 3 first one, but also inpatient, start happening a big outbreak in medical staff. 18 of 119 infected individuals in this hospital outbreak are medical staff, and we have a very high case fatality ratio of 15 deaths. So this outbreak really shocked the country, because it was the first big outbreak of SARS-CoV-2, especially involving large number of medical doctors and staff being infected. So just to summarize now what we have here, we have a whole hypothesis how by our detective work, how that happening. Again, by the 9th of March, a patient in the emergency department transmitting to another patient number 3, that start moving between wards, and then you start launching what you call outbreaks in different wards. So here we can have a very clear what you call putative transmission chains. Some of that are very, very, very sad. So for example, when getting the dialysis unit of 1, 2, 3, 4, 5, 6, 7 patients, 5 die within 3 or 4 days. It was a very traumatized program. In addition of major outbreak in the hospital, also causes a small chain of transmission in nursing home. So what we do, we get samples that we can, and now we have to see if we're going to support or refute of this hypothesis. So we would support this hypothesis if you have one virus, that is being transmitted between all the individuals, which the alternative hypothesis is what the hospital management believe, is that this is not a hospital outbreak, but that this is individuals that are coming to the community, both nurses and patients, and it is bringing that to the hospital. So we generate many genomes, around 20 genomes that we had samples from that, and we put on that time in relationship with samples of the environs. These mean samples also sample in Durban, that's where it happened, with the global diversity of the virus. At that time, the virus had very little diversity. But what we found of the 17 sequences that we have sequenced from the hospital outbreak, all of the 17 clustered together with very, very low genetic diversity. To be honest, they only add either 1 or 2 mutations between them. Really supported this change of transmission that entered the hospital and then with one unique source and causing an outbreak. So this what we do, we decide to produce a very detailed report. That's a report that it is around 45 pages that really report how this happened, the nosocomial outbreak, but also more important, how it is also caused the outbreak, but also with interventions could help to stop transmission in hospitals. And that's where it become one of our big suggestions: divide the hospital areas in green, yellow, and red areas without movements between patients, between red to yellow or yellow to green, and isolated staffs. And then we go to a large program in South Africa, and that we train over 10,000 medical doctors and nurses on how to understand and avoid hospital outbreaks. This report is also downloaded 100,000 times by 150 different countries, and become quite an important report in the beginning of the pandemic on how to understand and avoid hospital outbreaks. So then we launch around, like we after this report, we convince our government to invest in genomic surveillance, and our Department of Science and Innovation at the South African Medical Research Council give us a award of around like $2 million. That's quite a lot of money to South Africa. And then we decide to set up like 9 different genomics facilities across the country to be able to do sequencing in real time. And as you're going to see, most of these facilities were in academic institutions, but most of them are next to this green box. This is a big laboratory from the government, what we call the NHLS, the National Health Laboratory Service. And the idea is that we could do genomic surveillance week with a low turnaround time and get samples close where they are diagnosed to move to the sequencing lab. From the beginning, South Africa decided not to focus on a percentage of sequencing, but what we decided to do is to do a random proportional selection of around 50 samples per different province. We have 9 in South Africa, and try to do that in a low turnaround time. What we found that from the first wave, and just to ground you, in panel A, we have in black the wave of infections. That first blurb in the beginning is the large hospital outbreak that we got involved. In red, we have the reproductive number. We know when it's above 1, the epidemic is going to grow. And in blue is the genomes that we have produced. So we produced the first genomes of the first cases, the hospital outbreak, and we start increasing as the number of infections start increasing in South Africa. What we found that we have hundreds, if not thousands, of introductions into South Africa. So and most of them would be around March and April, and then most of them would be by people that have been to Europe with a European ancestry. Not surprising. That's where it is the epidemic was happening around late February and March, and around 80% of the flights to South Africa from overseas come from Europe. But we also find that these introductions that's here in panel C in black very early start causing super spreading events. So in our first wave, we got over 99.9% of the infections in South Africa by being localized and transmitted. And these infections, each time that they cause this super spreading event, what we find that many of them fix lineages. So as the title of the paper say, we have identified 16 novel lineages of SARS-CoV-2 in South Africa, and that we work with the Pango to get naming to them. So just to ground you, that's the first year of the pandemic in South Africa in 2020. In the x-axis, I have the date. In the y-axis, I have the proportion of the genome. And when I have white here, in reality, it's not that I do not have data. It's the opposite. Normally, I have 20, 30 different lineages. Some from South Africa, overseas. And then what we start seeing around in May, we start seeing the kind of spreading of the specific South African lineages, which the first wave that's between June, July, were kind of co-dominated by 3 different lineages that they got this numbering, B1, 154, B156, and 61. But everything changed in October, November, with one lineage seemed to emerge and take over. How do you discover that? That became known as the beta, was from some of the thousands of clinicians and nurses that we train when they notice in a hospital in the Nelson Mandela Bay unusual cluster of infections, and they contact us and we get samples, and then what we have to do there is to make sure, is that a hospital outbreak, a cluster of infection, or these are widespread variant. And what we do within a week or two, we sample 100 clinics in close proximity to that hospital, and what we found is that despite whatever we sequence, it's only one lineage, which become to know initially as the 501v2 and then the beta. The beta variant has mutations where we didn't want to have. Yeah, this means a lot of mutations in the spike gene, more specific in the receptor binding domain. One of them was very important, was the 501Y. When we identify this lineage, we present to the World Health Organization. That's what mutation that I marked quite strong, because that was also in some of the minks farms, yeah. And we discuss a lot is that animal reservoir, and I believe that was by looking for that mutation that allowed the UK to also identify the alpha. So the first three variants, alpha, beta, and gamma, they all have the 501 mutation. They also have another one that's much more important, the 484Q, that's been shown to really decrease neutralization, and then also mutations in the non-terminal domain, this blue area, which is like antigenic supersite, yeah. Once we detect a new variant, we very quick, we have a process of communication. We communicate that to our chair of our Ministerial Advisory Committee, and we have a call with the Minister of Health. At the same time, normally in the same day, we inform the World Health Organization Virus Evolution Working Group and the Africa Center for Disease Control. We found that's very important. If both our continental and international public health agents have knowledge, they know how to respond, yeah. In that case, if our minister finds that's important, he requests a presentation to, in South Africa, is the National Coronavirus Control Council, that is chaired by the president, our president Ramaphosa, and have all the 28 ministers. In that case, the president found that's important to go public, so he asked a press briefing to be organized by the Minister of Health and the Africa CDC, which we present our results. And normally the president follow within 48 hours in live TV, yeah, increasing the response level of South Africa. Why that? Because it takes around 48 hours to pass new legislation. We did that process many times when we detect the beta, when we detect the delta, when we detect the Omicron BA1, BA4, BA5. So it's a process that we got quite good on doing, and that was quite important in South Africa, this trust from the government and being transparent with the result, yeah. But the most important thing to us when we detect this variant that has clear mutations that seem to evade immunity is to answer the next big questions. And the next big question was, what's the effective of the vaccine? For that, what I did, in addition of leading the Network for Genomic Surveillance, I launched another consortium, what we call the Variant Consortium in South Africa, that involved 500 scientists, yeah, more or less. And that what we would do, that we would use that information, we would meet weekly, yeah, to try to answer the most important questions related to a new variant. That's the vaccine effectiveness of diagnostic, pathogenic, and also basic neutralization immunology. Yeah. So the first question that we had to answer, and that had to be done quite quick, is the beta variant, that time was known as the 501v2 or the B1351, escape from the vaccine that we had in South Africa. This means the Chadox, Oxford, AstraZeneca. We had a phase 1, phase 2, so what we did, we have to answer one quick question. Does plasma collected from people after receive of the Chadox vaccine neutralize the beta virus? Yeah. What we do that, our lab receive all the samples and do all the genomes to discriminate between beta and wild type, and then with colleagues in the what we do, generate pseudovirus that we feed the mutations, and also give the samples of the variant to another colleague that will grow the live virus. And what we found is that on the beta was a very strong attenuation of neutralization, with 79% in the pseudovirus and 60% on the live virus have what you call non-detectable neutralization. And that was quite important result. Of course, that the main results of a clinical trial should not be in the lab, but should be what happened in the clinical trial. In the case on the beta variants and the Chadox vaccine, that's a Kappa-Meyer of the clinical trial. That's a paper that we got with Shabi Ahmad in the New England. And what it shows that over time, yeah, we have like more infections than in the vaccine and the placebo. Yeah. So what was the problem of this trial was two or three. The first one, we didn't at that time of the pandemic, end of 2020, we still thought that the vaccines would avoid infections, which is not the case, and we know that very well. But the problem that this trial was only involved young people, which had very mild to moderate symptomatics. With another colleague, the chair of the Ministerial Advisory Board, what we did is to put together all the work done in South Africa and overseas on pseudovirus and live virus plaque assay to try to see what would be the vaccine neutralization impact by beta by all the vaccines that has been kind of approved in South Africa. So what we saw, all of them, we have a decrease of neutralization, not as extreme as the AstraZeneca, some of them much less, such as the Johnson & Johnson, Pfizer. And if you link together with clinical trials that had happened in South Africa during that time on Johnson & Johnson and Pfizer, we have very clear results that showing that the Johnson & Johnson and Pfizer have very high clinical efficacy against hospitalization and severe. The AstraZeneca was proven still to have a high clinical efficacy against hospitalization severe, but that came months later, and that data did not exist. And our government decided to roll out the Johnson & Johnson and Pfizer, which we still don't know if was the right decision. But that's one thing that the pandemic show when have to take decisions. And in South Africa was quite sad. They decided to donate the 1 million doses that they have to the AstraZeneca to other African countries. And that then they start with a very large trial called CISONC on the Johnson & Johnson of 500,000 healthcare workers, which give quite a nice paper in Lancet that show that very high effectiveness. So our network for genomic surveillance in South Africa, continuously doing a genomic surveillance during the pandemic. In South Africa, we have this very distinct wave. So just to ground you, here is the first wave in 2020, gray, that we had like 16 lineages in South Africa and multiple lineages. Then we had a very extreme wave dominated by beta in the end of the year of 2020. We had a very extreme also delta wave, and then when we think that everything has finished, we can start seeing a very fast increase of new infections, which become known as the Omicron. By then, we had quite an effective way to detect and respond to variants. How we did that, we discovered Omicron when we almost had no cases in South Africa. What's happened, alert was raised by one of the labs on 15th of November, showing that he had a S gene target failure. So that's one of the targets fail. That's normally the deletion 6970. They send the samples to us. We produce a genome in around 4 or 5 days. When we look at the genome, they all highly mutated and all the same. Then what we do, that's where we activated the network on around 22 of November. So what we do, once you look at that, what we do, it is that now that's in Gauteng, close to Johannesburg, so we do the same as we did in Beta. We get a look where the samples come from. We make a big ring around, and then we ask the lab to move around 100 samples from 100 different clinics, 100 kilometers apart, to our lab. Our lab's the one that can do much quicker sequencing. And we managed to move these samples. It's around 800 kilometers away. They come by plane in less than 6 hours, and we go to sequence very quick. We use a very fast sequencing process of nanopore technologies, and we can generate close to like 70 high-quality genomes within less than 24 hours. Which then now we are sure that this is a new variant, widespread everywhere in close to like 70 or different clinics. Then that is on the 24th of November, we work with the PANGO to get a lineage designation. We talk with our Minister of Health and our President on the 25th of November. They decide to do a press briefing. The 26th a variant is identified. So in South Africa, that's really validated. Before, you only had like one sequence from Botswana. It is that when we get a look on from the 26th to the 25th of November, 2 days, or if you put since we got the first genome, 72 hours between identification of the first sequence and completely validation and communication of that to public health authorities. Yeah. Again, we have to answer another crucial question: is this new variant Omicron? What's the effect of the vaccines? Yeah, here we did quite a very smart experiment with a colleague of us, Sandile Selle and Alex Sigal, that we tried to affect not only what would be the effect of the vaccine, but what would be the effect of booster on this variant. Yeah, in South Africa we didn't have people boosted, so we create a proxy. So for example, here in the orange dots is people that have two vaccinations of the Pfizer. The Pfizer is also known as BNT162, yeah, and then people that have been dual vaccinated and previously infected as a proxy of a booster, because we know that these individuals have a higher level of antibodies, yeah. And what we show is that Omicron, we had a decrease of antibody levels, yeah, but when we calculate vaccine efficacy, we were quite confident that with a booster of people previously infected and twice vaccinated, the vaccine would still have high efficacy. And we thought that these results were very important in South Africa to vaccinate our healthcare workers, roll a booster program, but also guide a whole lot of the world. Yeah. Other results that became very important, it is the other arm of the immune system, and we did that with some colleagues from University of Cape Town, yeah, and here to see what's the effect of the T cell. And what we found that the T cell is much more stable than the antibody. Just to ground you, here we have people with ancestral strain T cell response with one dose of adenovirus 26, that Johnson & Johnson, or 2 doses, you have a decrease on T cell response, but very little. The same with Pfizer, 2 doses, and also convalescent, this means previous infection. So what we saw is that close between 70 to 86% of the CD4 is preserved, and that information was quite important, and we think that's one of the main reasons why, together with vaccination, previous infection, and both antibodies and T cells, that Omicron was much less deadly, especially in South Africa. In South Africa, because we detected very early, we had very little cases, very fast communication to the government. We also made sure that our hospitals were well stocked with oxygen and healthcare workers, and we end up with a very, very very low mortality rate of the Omicron wave. Yeah. Despite of really quick identifying the beta, the Omicron, give some information allowing the UK to identify the alpha, generating all this data on vaccine neutralization, yeah, what we found is that despite South Africa really leading the science in the globe, we are punished. And we are punished, especially with the beta, our travel ban was 281 days. You don't imagine how long that is for a country that's in the end of the world. After South Africa, we only have Antarctica, yeah, but they punish us again with Omicron. Very fast response, and then we had like this completely block out of South Africa from the rest of the world. And we highlighted that this travel ban would be ineffective, because we know that this is a very highly fast transmitted virus. Also, we don't know if Omicron originated in South Africa. We just detected here very fast. Would be ineffective, yeah, would be unethical, but also quite important, that could damage the trust of public health in the globe. So countries, when they detect new variants or new pathogens, would be reluctant to be punished. And that's what's happened. Despite a severe travel ban, Omicron spread everywhere in the world, yeah, and caused the biggest wave of infection during this pandemic. Yeah. In spite of that, we did again the same. Yeah, we are quite thick-skinned when we detect 2 new sublineages, the BA4 and BA5. We also highlighted that very early to the world, and that the BA4 and BA5 is where went to dominate the next wave of infections. They were quickly identified in South Africa. We can now generate all this neutralization data from both Omicron, BA4 and 5 in less than 15 days. So we have quite effective way identify variants, understanding the effect of the vaccine, understanding how much transmissible. And despite being punished the other time, we decided to go public again, and I think that the world learning a lesson that they haven't punished South Africa for the discovery of BA4 and BA5, which all the main sublineages, for example, the the BQ, it is a sublineage of the BA4 and 5. Yeah. Another thing that we really did, we thought that would be unethical to us in South Africa to keep this technology that we have and affect the genomic system in KRISP and SERI, just to South Africa. So we work with the Africa CDC and the World Health Organization to create what it's called the Africa PGI, the Pathogen Genomic Initiative. And in this initiative, you have 3 levels of laboratories. You have the national sequencing labs, you have the regional labs, and then you have the specialized genomics facility, which KRISP it is, and SERI it's one of 2, the other one's in Nigeria. And the main objective of this specialized genomics facility, it is to produce data for other countries, but also doing training. And in our labs, we produce data for 26 African countries in real time, really to try to use this to generate genomic surveillance in Africa. Our team also got involved in a holistic collaboration. What's a holistic? That we decided that we would do different than a lot of times collaboration as happened with Africa and Latin America, that someone use the data, analyze the data, and then after present a whole paper and may even add auto. So what we did is like meet every week and came with the scientific questions together, analyze the data together, and then once the paper was finished, the submission, we would give all the analysis scripts and we would train all the African scientists on the analysis of the data. That worked quite well. We got the first paper in Science on the first year of genomic surveillance, another one a few weeks ago, and that involved hundreds of African collaborators of 53 or 55 countries, and really showing that we can work together to really understand what's happening in the continent. Another thing that we also did a lot in our lab, and that was very important for my own staff, it is to give back. To give back on training, and we have received like hundreds of fellows in our labs, for training from many African countries. That is the main job of a specialized genomic facility. And here was an interesting workshop that we did. We did dozens. We did 26 of those workshops, during the pandemic. But this one, we are receiving people from 17 African countries. And when I get a list, the name of the list of African countries, we have some of the most resource limited countries in Africa, like São Tomé and Príncipe, Comores, South Sudan, Mozambique, Angola. So I decided that would be unfair to take them to our labs. Our labs are very, very rich and developed. So I challenged my team to put all the reagents and equipment in a small car. We use a small micro and transfer that to a normal seminar room. And the effect was fantastic, because when a lot of these fellows came, and they saw how little equipment you need to do genomic surveillance, they really trusted that they could do in their own country. And in the end, 15 of the 17 countries that participate within weeks start generating data. So what we also do, we invite countries to send fellows to spend weeks or months in our lab. In total, we had 233 African fellows that were trained from 39 African countries during the pandemic. And that was really exciting. But that's something that we didn't do only in the pandemic. We have hosted over 2,000 African fellows during the last 15 years that I have been running a genomics facility in South Africa. So what they normally do, they come, in the case of the SARS-CoV-2 and during the pandemic, we normally have the samples before, and then we would train them on producing data in their own samples. And it was quite exciting. We had to kind of send them in the night back to the hotel they went to stay in the lab. And it was really exciting, not only for them, but also for our own people, because that we are in very big pressure, but they made many different friends. We have got many awards and accolades. To be honest, we are not chasing that. One of them that I'm quite proud It is the time 100. Why I'm proud of that? We also in this week we also got another award called the German Africa. Is because I shared that with Dr. Sikolile Moyo, and Sikolile was a PhD student of mine for 4 years, and now he's a lab director of Botswana Harvard. And so exciting to see like the previous students have now graduated and now become leaders. Yeah, so I'm very proud of that kind of recognition, especially because just showing that our long-term capacity building is beginning to pay off. Yeah. Another thing that we became really involved in South Africa is on public communication of science. We thought that that was really important, yeah, because we had so much fake news on the internet, and we devote a lot of our time. We made 8 times the front page of the New York Times, over 10,000 articles, that a readership of close to 3 times the world population, and that's something that was very important to try to balance the fake information. Yeah. So as a highlight, many of these papers we wrote after was not the main objective, but because the papers were quite impactful, because they guide a lot the response, they end up in some of the top journals in the world, and we got over 30 of these papers like in the top journals. What's quite good for the scientific career, especially of young African scientists. So just to conclude, yeah, we in South Africa we had previous experience dealing with epidemics, especially on virus and respiratory pathogen like TB. But my own group on emergent viruses, we participate with colleagues in Brazil and Latin America of the genomic surveillance of Zika, dengue, chikungunya, yellow fever. So we are quite used to do that. Yeah, we prepare quite early. Once we discover a variant, we grow the genomic surveillance to involve large working groups of collaborative work, so from diagnostic, clinical, epidemiology, immunology, and basic virology and vaccines. Really important not to isolate genomics, but really genomics should only be a tool to answer the important questions. Yeah. We are very persistent. In South Africa we have been discriminated forever. We have very thick skin, and we are used to fight discrimination and difficulties, and that help a lot, especially about a lot of us being HIV/AIDS scientists. We are kind of activists, and we don't bow easily to pressure. Yeah. We had a very clear line of communication with government that follow and acted of the science about variants, yeah, in our own group to give back to the community, yeah, with training medical care workers and scientists. That was quite important. That helped us to also identify variants, but really allow our people to hold the pressure, make friends, and have fun in the way they thought that they were contributed for a greater good in the African continent. Yeah, we are back to neglected epidemics that have been neglected in the last few years. So we are doing a lot of genomic surveillance for HIV, AIDS, and TB, and we are trying to launch what we are very excited with Brazil, Latin America, and many of our global partners. Yeah, a new large consortium that we call Climate, to deal with climate amplified diseases and epidemics. We are really excited about that, and would be great if you would like to engage with us on that. Yeah, thank you very much, and apologies that I'm not there in person, but I trust that you're gonna have a great virus genomics and evolution workshop.